Okay, good morning everyone. Okay, good morning everyone. Please confirm me, am I audible to everyone? Good morning Shabina. Just confirm me, am I audible to everyone? Good morning, just confirm, am I audible to you? Okay, please confirm me, am I audible to everyone? Good morning, good morning. Perfect. Okay. Let's wait for 1 minute, let others also join very quickly. Very good morning. Wonderful. Sudarshan, I believe you're going through the recorded classes strictly, right? Please don't miss any kind of a class. Watch the classes compulsory. Very good morning to everyone. Okay, friends. Now let's try to understand other cell organelle. So, if everyone very quickly recall, friends, so far what kind of organelles that we have completed, after the plasma membrane, we have started reading about the nucleus. Then we have read about the mitochondria. After the mitochondria, in the yesterday class, friends, we have understood about a different type of the plastids. And after the plastids, we read about the peroxisomes. Correct, na? These are the various type of the organelles that we have studied actually. Now, friends, now we are going to cover a very important organelle among all, the lysosomes. Okay, there is 60 to 70% of the chances are there, friends. We can expect the question from this particular topic of the lysosomes. I will explain very slowly to all of you. I want you to first focus on it and understand how to read it. Lysosomes. If I ask you what is the major function of the lysosome, you all people read from the 10th standard, we all use it to say one common statement. Lysosomes are the suicidal bags. Correct now? Why? Just give me 1 minute. What do we call, friends? Lysosomes are said to be the suicidal bags. Now the question is here, why we use it to say lysosome is the suicidal bags, and sometimes, friends, we can say that lysosomes are garbage disposal. See, friends, in general, what happens inside the cell whenever cell become older, whenever the cell become older, what has to happen, friends? The old cell has to become die, and this cell has to be replaced with the new cell. What, friends? The old cell has to die, and it has to be replaced with the new cell. Sometimes what happens, friends? This particular old cell, which is non-functional, cannot be replaced with the new cell. At the same time, friends, this old cell never ever die also. That time what is going to happen, that cells remain as it is old, and there is a possibility that they undergo the continuous division, and there is a possibility that they can cause the cancers. Those kind of cells can be digested completely by the lysosomes. No need to get confused over there. So, friends, which substances we can call it as garbage inside the cell? The substances which are no longer required for the cell. Everyone, please continue. Good morning. The what I'm trying to explain you, friends, the substances which are no longer required within the cell, we generally call it as the garbage, and that garbage can be cleaned completely by this particular lysosomes. Now, friends, listen everyone very carefully. When I say the lysosome, remember, friends, lysosomes can digest almost any kind of a molecule. Good morning, good morning. Please continue, everyone. Lysosome can digest almost any type of a molecule. What does it mean? It can digest the lipid, it can digest the protein, it can digest the carbohydrate, it can digest any kind of a molecule very, very easily. Now, why lysosome can digest any kind of a molecule? The reason is that lysosome contain abundant amount of the hydrolytic enzymes that are having the ability or capability to digest any kind of a molecule. Suppose example, everyone pay attention for 20 minutes continuously. Suppose example, friends, if you see the enzymes nucleases, what is the major function of nucleases, friends? They are generally involved in cleaving the phosphodiester bond within the in simple words, they are involved in cleaving something inside the nucleus, whether it may be endonuclease or whether it may be a hexonuclease, which are involved in cleaving the phosphodiester bond especially. Then the next one we can call proteases. You know very well, proteases are generally involved in the cleaving of the peptide bond. Then friends, what we can call glycosidases. Glycosidases are involved in the cleaving of glycosidic bond that is formed within the sugars. And what about the lipases, friends? Lipases are the one which are involved in the cleaving of the ester bond. And what happened? Phosphatases. You know very well, phosphatases are those type of enzymes, friends, that plays a key role in removing the phosphate. The next, sulfatases. What is the major function of sulfatases? Which are involved in removing the sulfur group. And what are phospholipases? Phospholipases are those type of enzymes, friends, which are involved in cleaving the phospholipid. So what I am trying to explain to all of you, the hydrolytic enzymes that are present in the lysosome, they can cleave almost any kind of a substance. No need to get confused. But here comes a very, very interesting point for the exam. Whatever the enzymes that are present in the lysosomes, friends, if that enzyme wants to be active, compulsory they require what, friends? Compulsory they require the pH which is strictly acidic in nature. That's why whatever the hydrolytic enzymes that are present within the lysosome, we called them as the acid hydrolysis. Can you see the picture, friends? Inside this particular lysosome, we are maintaining the pH hardly 5. Now the question is here, how can we maintain the pH acidic within this particular what we can say within this particular lysosome? Can you see the picture very clearly? On the membrane of the lysosome, there is a presence of H plus ATPase pumps. You know basic function of the H plus ATPase pumps. By the ATP hydrolysis or by spending the ATP, what they are going to do, friends? They are pumping the H plus ions continuously into the lysosome. Because of the continuous influx of the cytoplasm within the lysosome, the pH will become completely acidic, friends. That's the reason only we call them as the acid hydrolysis. Do not get confused. Now, all the students has to get a brilliant doubt. That is, suppose example, all the hydrolytic enzymes that are present within the lysosome, what happens if they enter into the cytosol? You know, friends, generally cytosol is having a neutral pH. Even though all the enzymes of the lysosomes entered into the cytosol, they are strictly inactive. They cannot perform any kind of a function. Why? Because if they want to be functional, compulsory they require what, friends? Acidic environment only. There is no need to get confused. Now, let me tell you one very interesting point. Understand 20 minutes continuously, friends. Don't deviate from the concept. Within the lysosome, listen everyone, within the lysosome, there are more than 150 hydrolytic enzymes are present. What, friends? Within the lysosome, there are more than 150 hydrolytic enzymes are present. Now, here comes a very interesting point. From where all these 150 hydrolytic enzymes are coming from? Again, most of the students will consider that all the hydrolytic enzymes are coming from the cytoplasm, which is absolutely wrong statement. See, in the last class, friends, we have studied mitochondria, we have studied chloroplast, we have studied the peroxisome, we have studied the nucleus. What you people remember here, all the enzymes got imported into these organelles exclusively from where, friends? Cytoplasm, correct now? But if you observe whatever the enzymes that are getting imported into this particular what we can say, friends, lysosomes, they are exclusively coming from the endoplasmic reticulum. Now I'm going to explain you very briefly. I want you to pay attention very correctly. Look at this. Understand, friends, the question won't go beyond the concept that we are discussing. Make sure that you understand the concept in a brilliant way. Lysosomes are the membrane sacs of the hydrolytic enzymes that digest both extracellular materials and worn out organelles also. Now, see this interesting point. All the hydrolytic enzymes are exclusively active at the acidic pH. Therefore, even if these enzymes escape into the cytosol, friends, they are not at all active at the neutral pH. Do not get confused. Now, friends, entry of the enzymes into the lysosome, we generally call it as sorting of lysosomal protein. What is the mechanism of sorting, friends? Entry of the hydrolytic enzymes into the lysosome. That is what exactly we can call it as sorting of lysosomal protein. I am going to explain everything very briefly and elaboratively. All the students just focus on the diagram very correctly. Don't confuse. If you understand this diagram very specifically, you get a crystal clear idea how the enzymes are entering into the lysosome. Friends, everyone listen, friends, this is the lysosome. Correct? Now, this is what? A vesicle. Now, don't confuse, friends. This is the Golgi complex. You all people know very well. This is called as the cis Golgi. And what about this? This one we generally call it as the trans Golgi. Correct now? Now everyone imagine my statements and follow me exactly. Consider, friends, this is the ER. Okay. Now what we discussed just now, there are more than 150 hydrolytic enzymes are going to enter from the endoplasmic reticulum into the lysosome via Golgi complex. Every student, please look into the picture, friends. See, there is a protein. Everyone understand? There is a protein or there is an enzyme entered successfully into the Golgi complex from the endoplasmic reticulum. So, friends, whenever this particular protein entered into the Golgi complex, in the cis Golgi complex, what is going to happen? Immediately to this particular enzyme or protein, mannose 6-phosphate will come and attaches. Now, every student has to understand why the mannose 6-phosphate attached to this specific protein. The answer is very simple. The mannose 6-phosphate generally acting like a sorting signal. What is the meaning of a sorting signal? This sorting signal helps this particular lysosomal protein to enter into the destined area, to enter into the targeted area. That's why mannose 6-phosphate will come and attaches to the lysosomal enzyme in the cis Golgi. And you know very well, it is going to travel various compartments of the cis Golgi. Once it travels into the various compartments of the cis Golgi, can you see the picture, friends? Finally, it will come and attaches to the receptor present on the trans Golgi complex. This is also extremely crucial point. Listen, friends. In the trans Golgi, can you people see there is a receptor is located? Listen, I'll explain twice. In the trans Golgi, there is a receptor is located. Friends, to this particular receptor, who will go and attaches lysosomal enzyme along with the mannose 6-phosphate. If every student observe the diagram very correctly, please don't think that Lysosomal enzyme directly attached to the receptor. No, that statement is absolutely wrong, friends. Lysosome did not attach to the hydrolytic enzyme. Sorry, hydrolytic enzyme did not attach to the receptor. Here, what you people observe, hydrolytic enzymes are associated with the mannose 6 phosphate. Is it or not? So, friends, actually, the receptor is act as a binding site for the mannose 6 phosphate. To the mannose 6 phosphate, hydrolytic enzyme is associated. Very good. Then what happened? This particular receptor, it will come out, which means what? It will be bud off. It will be bud off from the trans Golgi complex. Friends, this is the question now. Whenever they bud off from the Golgi complex, everyone please try to understand, they are covered, they are protected by a specific coat. What is the name of that particular coat? We call it as the Clathrin coat. This is also a question you people can expect the question in the exam. What is the name of the protein that acting as a coating or a coat to the vesicle? Now you can ask me, sir, what is the major use of this particular Clathrin? Suppose, friends, this is the vesicle. It is transporting within the cell. Whenever it is transporting within the cell, it can be prevented from the degradation with the help of the Clathrin coat. Not only that, friends, it will provide greatest stability to the vesicle. What? Clathrin coat provide the greatest stability to the vesicle. At the same time, Clathrin coat will prevent the degradation of the vesicle. Now, every student, please look into this. Then what is going to happen, friends? This particular vesicle will travel with the help of a cytoskeletal apparatus, that is motor proteins such as dynein, kinesin. Finally, they will go and fuse with the lysosome. Can you see the picture? Whenever they will go and fuse with the lysosome, friends, what is going to end? What is going to happen? Hydrolytic enzyme receptor along with the mannose 6 phosphate entered into the lysosome. I believe you all people remember, in the lysosome, what kind of a pH we can maintain? We can maintain the acidic pH. Because of the acidic pH, what is going to happen, friends? Mannose 6 phosphate along with the hydrolytic enzyme will be separated and receptor also gets separated. Then at the end, what is going to happen? There is a removal of the phosphate group, and finally, friends, the lysosomal hydrolytic enzyme completely become matured. There is no need to get confused over there. Now, what happened to this specific receptor? That is also extremely important, friends. This particular receptor, listen everyone. This particular receptor again packed. Can you see that, friends? Again packed from the lysosome, packed in the form of a vesicle, and that vesicle will come and fuse to which complex? Trans Golgi complex. Now, why this vesicle come back and fuse with only trans Golgi complex, friends? Because we have to place the receptor only on the trans Golgi complex. There is no need to get confused, friends. It is very easy to understand. Let me summarize one more time very clearly to all of you. Just focus on the picture very clearly. As we discussed, the lysosome contain, there are more than 150 hydrolytic enzymes. Since it contain more than 150 hydrolytic enzymes, what you people has to observe here, all these 150 hydrolytic enzymes are exclusively synthesized in the ER. From the ER, they are reaching the lysosome via Golgi complex. There is no point of getting confused over there. Initially, what is going to happen, friends? From the endoplasmic reticulum, this particular enzyme entered into the cis Golgi complex. Friends, once it entered into the cis Golgi complex, what happened? In the cis Golgi complex, to the hydrolytic enzyme, mannose 6 phosphate will be added. And I told to all of you very clearly, friends, what is the major significance of the mannose 6 phosphate? Mannose 6 phosphate majorly acting as signal sequence. So what it will do? It will help this particular hydrolytic enzyme to travel throughout the Golgi complex and to reach up to the lysosome. Then what is happening? this particular lysosome enzyme along with the mannose 6 phosphate travels all the compartments of the Golgi complex. Finally, where they will come and reach? Finally, they will come and attaches to the receptor present in the Golgi complex. I am explaining you one more time. This receptor is not at all for the hydrolytic enzyme, friends. This receptor is specifically acting as a binding site for the mannose 6 phosphate. Later, what is going to happen, friends? Later, this entire vesicle can be surrounded by a clathrin coat. Can be surrounded by what, friends? A clathrin coat. Now understand my statements very correctly. Now what is the major function of the clathrin coat? It prevent the vesicle from the degradation. At the same time, it increase the stability of the vesicle. And friends, finally what is going to happen? The vesicle will directly come and fuse with the lysosome. Once it fuses with the lysosome, hydrolytic enzyme mannose 6 phosphate along with the receptor enter inside. As we already read, inside the lysosome the pH is acidic. Because of the acidic pH, receptor is getting separated and lysosomal enzyme along with the mannose 6 phosphate is also getting separated, where there is a removal of the phosphate group. Why we are removing the phosphate group? Just now we studied that the lysosome contain phosphatase enzyme which generally remove the phosphate group. Then friends, what is going to happen? It immediately become matured. And what about the receptor? This receptor again completely cycled back. Again it is packed in the form of a vesicle, and that particular vesicle come back to the trans Golgi complex, and it is going to express on the membrane of the on the trans Golgi complex, and it is further waiting for the another hydrolytic enzyme. Friends, this is called as the biogenesis of the lysosome. In other words, how the lysosomes are getting transported from the endoplasmic reticulum to the lysosome. Every student, please try to understand the steps very clearly. What is the first step, friends? There is the addition of the phosphate group to the mannose residue. That is what we read. And listen, this because you can expect a direct questions in the exam. And what is the significance of this particular mannose 6 phosphate? This mannose 6 phosphate forms a sorting signal that moves through the cistern to the trans region, where it binds to where specific receptor. Why only it is moving to the trans region, friends? Because the trans Golgi complex only contain the specific receptor. No need to get confused over there. Now, after it binds to the receptor, generally what is going to happen, friends? Just now we discussed that entire receptor can be surrounded by a coat. What is the name of that particular coat, friends? The name of that particular coat is called as the clathrin coat. There is no point of getting confused over there. Good morning, good morning. Very good doubt, Hannan. Very good. It is having a higher affinity towards the mannose only. Very good, valid question. Then what happened? It moves away to fuse with the late endosome. After fusing with the late endosome, what is happening, friends? Very simple. The phosphate is removed. Entire hydrolase enzyme dissociated from the receptor. Phosphate group is also removed. Then automatically the hydrolytic enzyme will become functionally active. And what happened to the receptor, friends? Receptor is getting recycled back to the trans Golgi complex in the form of a vesicle. This is how, friends, the lysosome protein synthesis will be taking place. I believe everyone understand very clearly about the protein synthesis, how the proteins are transporting from the ER to the lysosome. Every student very quickly confirm, is it clear? Quickly confirm, friends. Is it clear? Every one of you quickly confirm, is it clear, everyone? Till the biogenesis, everyone clear? Great. I hope it's clear for everyone. So friends, so far what we have studied is just how the proteins are getting synthesized within the lysosome, and how the proteins are getting transported. Everyone has to respond. Come on, please be active. And friends, what are the different types of the lysosome? Now we have to read types of the lysosomes, functions of the lysosome, and most importantly, friends, we have to read all the diseases that are associated with the lysosome. That is what we have to read. Number one is called primary lysosome. Number two is called secondary lysosome. Number three, phagolysosome, autophagolysosome, and residual bodies. That's a very valid question. All very good, very good. Now listen, Hanna. There are more than 150 hydrolytic enzymes, right? They are all they never be transferred at a time. Mostly they will be transferred in a sequential manner. Even though they transported at a time, there is no problem. In the cis Golgi there are many type of mannose receptors are sorry, in the trans Golgi there are many mannose 6 phosphate receptors are there. They can accommodate most of the hydrolytic enzymes at a time also. There won't be any problem, but most of the time the transport occur sequentially only rather than simultaneously. Now friends, let's try to understand what are called as primary lysosomes. Listen, here understanding the definitions are very easy. Okay. Now, what are primary lysosome? Friends, just now we discussed. Every student, please focus on the diagram without having any confusion, friends. Every one of you. This is a endoplasmic reticulum. And what about this, friends? This is called as the Golgi complex. Correct na? So the protein got synthesized in the endoplasmic reticulum, travels through the Golgi complex. See this, friends. See this, friends. And this is the vesicle, final vesicle. The vesicle which contain the abundant amount of the hydrolytic enzymes. Friends, interestingly, even though the vesicle contain abundant amount of the hydrolytic enzymes, friends, all the hydrolytic enzymes are absolutely inactive. Now, the most important statement that every student has to remember for the exam, why all these hydrolytic enzymes are absolutely inactive? Why? Because friends, they have not encountered to any kind of a pathogen or any kind of a food particle to be digested. Since it is not reacted with anyone, all the enzymes which are present in the lysosome absolutely inactive. So friends, how can I define this definition now? Listen, primary lysosomes are newly formed by budding from the Golgi complex, and therefore they have not encountered any substrate for the process of the digestion. That's the reason, friends, we called as all the hydrolytic enzymes are inactive. That's the reason primary lysosomes are generally said to be what, friends? Just storage granules. They're just called storage granules because hydrolytic enzymes are inactive. And why the hydrolytic enzymes are inactive? They have not encountered with any substrate. They have not encountered with any pathogenic particle. That's why they're absolutely inactive. Now use the brain. Every one of you, please try to understand the significant difference between the primary lysosomes and secondary lysosome. Listen, friends, this is called as macrophage. What is this? This is called as the macrophage. I believe everyone remembered what is the major function of the macrophage in the immune system. You know very well, whenever a pathogen enter in our body, immediately macrophage will trap that pathogen. Friends, everyone, please look into this picture. This is the pathogenic particle. What happened? It can easily trap that particular pathogenic particle and brings that pathogenic particle inside. Whenever it brings the pathogenic particle inside, what do we call this structure, my friends? I believe everyone remember. We called it as the phagosome. I want all of you to understand me correctly. Whenever a pathogen enter in our body, immediately macrophages trap that particular pathogen, and that particular pathogen trapped in the packed in the form of a vesicle, and it brings inside. Then it is called as phagosome. Why it brought inside? Because inside we have the lysosome enzymes which are synthesized just now and which are present in the inactive form as the storage granule. Now tell me friends, what is going to happen now? The lysosomes and phagosome will combine with each other. Then we get in a structure called as phagolysosome. What the phagolysosome will do, friends? The lysosome will release all the hydrolytic enzymes. All those hydrolytic enzymes will go and destroy that specific pathogenic particle. No point of getting confused over there. So once again, everyone listen. Secondary lysosome resulted from the, because friends, I want you to remember the definition as it is without having any confusions. Secondary lysosomes are formed from the repeated fusions of the primary lysosomes with a phagosome. Correct now? So friends, we never called the secondary lysosome as the storage granules. Why? Because all the enzymes become absolutely active right now. That's the reason, friends, we called them as the digestive granules. There is no need to get confused over there, and it is very easy to understand. So friends, you understand what is called as primary lysosome, you understand what is called secondary lysosome. Primary lysosomes, they are just acting like a storage granules because they're just synthesized and they have not encountered with the any pathogenic organism or any other substrate. That's why they simply act like a digestive granules only. But when we consider the secondary lysosome, they will fuse with the phagosome which trap the pathogenic particle. That's why they never act like a storage granules. So all the secondary lysosomes are acting like a digestive granules. There's no point of getting confused over that. Next one, we call it as the phagolysosome. No point of confusing, friends. Phagolysosome is a kind of a, see, understanding the concept is very correct. How does this particular phagolysosome is formed? Just now we discussed phagosome combined. What is the function of phagosome? Which traps the pathogen. Phagosome combined with the lysosome which contain the hydrolytic enzymes. Then there is a formation of what, friends? Phagolysosome. And most important point, autophagolysosomes. This is the most important statement for the exam. And for this only in the 2017, the Japanese scientist awarded Nobel Prize. Okay, autophagolysis. See, friends, you know, right? What is the general biological process has to happen inside the cell? You know, what happened inside? Cell will born, cell undergo the division. Correct, na? At one particular point of time, cell will stop the division. Upon stopping the division, friends, cell will undergo the process of differentiation. After the differentiation is over, the cell is going to perform the function. After the function, the cell has to die. Whenever the cell will die, friends, automatically it is replaced with the new cell. This is the most common biological process has to happen in every organism's body. But sometimes what happens, friends, even though cell become older, even the cell become older, they never ever replaced by the healthy cell. Then what happened to this particular old cell? Anyway, friends, it is not at all functional. First point, it is not at all functional. Second point is that it is not at all getting replaced by the new cell. So there is a possibility that it may convert into the cancerous. That time what is going to happen? This particular autophagolysosome is going to destroy its own cell. What, friends? The autophagolysosome is going to destroy its own cell, which is not functional. That's the reason only, friends, we call it as the autophagolysosomes. There is no need to get confused over there. It is a kind of the secondary lysosomes which are involved in digesting the organelles which are completely aged. No point of getting confused. So friends, listen very carefully. Understand. The question won't go beyond the PPT level. So make sure that you'll understand very correctly. We clearly understand what are primary lysosomes, which are just storage granules. And next one is secondary lysosomes, which are acting as the digestive granules. And the third one we can call phagolysosome, which is formed by the combination of or fusion between the phagosome and lysosome. And just now we discussed autophagolysosome, which is generally called as the suicidal bags, because it is going to digest the cell which are aged. Now the last and final one, don't confuse friends, this is also very important. I want you to remember for the exam. One is called as residual bodies. What is that? Residual bodies. There is no point of getting confused over there, and it is very easy to understand also. What is called as residual bodies? Friends, look into the a very simplest definition first. Later I will explain you more depth. Focus, friends. Residual body is nothing but secondary lysosomes with undigested waste. Now, first, tell me, friends, what are secondary lysosomes? I already told you secondary lysosomes are type of the phagolysosomes. What are phagolysosomes? We have studied. This is the phagosome combined with the lysosome. This is the phagosome combined with the lysosomes, and this is what we can call it as the phagolysosome. What is the function of the phagolysosome? It is going to digest that particular pathogenic particle, or it is going to digest other substrate. Whenever it is digesting the other substrate, whatever the substrate or the particles that are undigested, what friends, whatever the substrate or the particles that are undigested, we call it them as residual bodies. There is no point of getting confused over there. Listen friends, don't confuse it. It's all about brilliant understanding. Secondary lysosomes with undigested waste, that is what exactly we can call the residual bodies. See friends, most of the time this residual bodies will be in the yellow to brown granules. Yellow to brown granules. These kind of a granules which are in the yellow to brownish color, we generally call it as a lipofuscin granules. You have to remember compulsory for the exam point of view. Listen once again. The second, come on, be active. The secondary lysosomes digest the content of the phagocytic or autophagic vesicles to form the residual bodies. Now you can, where this particular residual bodies are most commonly found, they most commonly found in the liver, kidney, heart muscle, retina, adrenal nerve cells, and ganglionic cells. Now, you can ask a very valid question. Sir, what happened to this particular residual bodies? Everyone understand my point correctly. 90% of the cases, I am using the word 90%. 90% of the cases, this particular lipofuscin granules undergo the process of exocytosis, which means friends, they can easily go out of the cell. And what about the remaining 10%? Remaining 10% of the residual bodies will be present within the cell only. Friends, even though they remain within the cell, they won't cause any kind of a damage. Sometimes they are automatically degraded within the cell itself. Every student, please look into the picture very correctly. All of you focus, friends. This is the lysosome. You can see in the picture so clearly. This is the phagosome, and sorry, this is the lysosome, and this is the phagosome. What is the function of that particular phagosome, friends? This particular phagosome trapped this particular pathogenic molecule. Now that time what is going to happen? The lysosome combined with the phagosome, it is going to form a autophagolysosome. Now once autophagolysosome digested the particles, if any particles are left over, that is only we call it as friends residual bodies. Most of the time what is going to happen, friends, the residual bodies, 90% of the cases I told to all of you, they undergo the process of the exocytosis. Very clear. And what happened to the remaining 10%? Remaining 10%, they will be retained within the cell only. Whichever is retained, whichever is present within the cell, those kind of granules only, friends, we call it as the lipofuscin granules, which are present within the cell only. Mostly they won't cause any harm to the cell, or sometimes it automatically degraded within the cell itself. There is no point of getting confused over there, and it is very easy to understand. Tell me, friends, what we have understood so far. Lysosome is a kind of a very big cell organelle, friends. Initially, we understand biogenesis of lysosome means how the hydrolytic enzymes are getting synthesized in the endoplasmic reticulum and how they are getting transported to the lysosome. Then we understand just now that types of the lysosome, that is primary lysosome, secondary lysosome, autophagolysosome, and residual bodies. And friends, what else is left over? Now we have to read all the functions of the lysosome. After the functions, most important for the exam point of view, diseases associated with the lysosome, that also we have to read and remember compulsory. Okay, now listen. Major function: heterophagy, autophagy, extracellular digestion, then followed by the autocytolysis. Friends, it is very easy to remember the function. You have to remember the definition along with an example if it is possible. Everyone, listen, friends. Again, you people consider, friends, this is a macrophage. I don't know how many of you, aware of the mechanism. Everyone pay attention, friends. What generally happen in the human body? What generally happens in the human body whenever a pathogen enters in our body? Let's see, friends. This is a pathogenic particle, consider a bacteria. Immediately, which cell will be active? Everyone listen. This is called as macrophage, which is present, which is completely wandering or moving in the blood circulation. Tell me, friends, what this particular macrophage will do? First, macrophage trap this particular pathogen packed in the form of a vesicle. What do we call it, that particular vesicle? That is only we can call it as the phagosome. And you know very well, inside the macrophage, what is present, friends? Inside the macrophage, lysosomes, that is hydrolytic enzymes are present. Now this lysosome and phagosome combined together, they can form the phagolysosome. They will break down that particular pathogen particle into very small, and they expressed finally on the MHC. They expressed where? They expressed finally on the MHC. Then immediately what is going to happen later? Our T helper cells will come, will take that antigen. They will activate the B cell. B cell produce the antibodies. Finally, antibodies will come and destroy. We all people know that very commonly. Correct na? That is what we read. So here, from where this particular pathogenic particle is coming out? Friends, this particular very good, Shabina. From where this pathogenic particle is coming out, it is coming from the external. That's why we call it as a heterophagy. There is no point of confusing, friends. Digestion of the material of extracellular origin. That is what we can call the heterophagy. No point of confusion. Lysosomes pick up the foreign invaders such as bacteria, food, and break them into small pieces, and they will be expressed on their membrane surface, followed by the activation of T helper cell, followed by activation of B cell, followed by the production of antibodies. Antibodies is going to destroy. That process of digestion of the material of the extracellular region, we call it as, friends, heterophagy. There is no need to get confused. And friends, it was not shown in the picture. Then what is called autophagy, friends? Heterophagy means we are digesting a material which is coming from the extracellular origin. And what is called as autophagy? Autophagy means destruction of the particle inside, means digestion of the material of intracellular region. I told you, na friends, sometimes organelles will become old, sometimes organelles will become non-functional, or cells become non-functional. That time we are going to destroy that particular organelles or the non-functional cells within the I mean to say within the cell itself. That process only we call it as the autophagy. It is picture is not showing here. So heterophagy is digestion of the material of extracellular origin. Autophagy is digestion of the materials of the intracellular origin. There is no point of getting confused over there. Next one, we call it as the extracellular digestion. This is the third function, third major function performed by the lysosome. I believe everyone remember, if you observe the structure of the sperm, you know that the sperm contain the head. And you also know very well the head contain what, friends? A head contain the nucleus. And what else it contain? It contain a propulsion system. And very important, friends, it contain an acrosomal vesicle. You know very well. This is called as what? Acrosomal vesicle. And you know very well that acrosomal vesicle contain abundant amount of the hydrolytic enzymes. There is no need to get confused over there. It is easy to understand also. Okay. Now, what is happening, friends? This particular sperm, with the help of a head, first it will go and fuse with the egg membrane. Whenever it fuses with the egg membrane, automatically what is going to happen? The acrosomal vesicle will be active. It is going to release abundant amount of hydrolytic enzyme. It can easily digest the zona pellucida layer present on the egg, and it can easily enter into the egg cytoplasm. This kind of a mechanism, friends, we call it as extracellular digestion. There is no point of getting confused over there, and it is easy to understand. Then, friends, the last and final mechanism, the final function performed by the lysosome, we call it as autocytolysis. This is the very interesting point. You know, friends, so far we understood that lysosomes are generally required for the what process? Lysosomes are generally required for the destruction. But you see the point here, first time, even though lysosomes are breaking down something, listen friends, even though lysosomes are breaking down something, friends, they are extremely useful in the development process. This is the unique point to be remembered for the exam. Suppose an example, friends, listen, this is the tadpole tail. What, friends? This is the tadpole tail. You know, no, during the time of the maturation, what is going to happen? This tadpole only develops into a mature frog. Now, the point is here, very good. How does this particular tadpole tail is getting breakdown? Or how this tadpole tail is getting digested? That is an important statement. Friends, this particular tadpole tail contain abundant amount of the lysosomes only. So what happens, friends? This particular lysosomes are digesting its own tail as the process of the development. That's why we call it as autocytolysis. Lysosomes may also be important in the process of the development. For instance, they are responsible for the breakdown of a tadpole's tail as the tadpole develops into a frog. As the tadpole develops into a frog. In this process, the lysosome release the hydrolytic enzymes where into the cytoplasm to digest the cell of its own. Since it is digesting the cell of its own, friends, we call it as the autocytolysis. These are the major functions performed by the lysosomes. Now, friends, last and final one, that is the diseases that are associated with the lysosome. There is a great possibility, friends, in the exam mostly they will write the question from the diseases. So that's why I want all of you has to focus on the diseases compulsory, because they can give like this. They'll give four organelles in one column, and they can ask the diseases associated with that specific organelles. Or in one column they use it to give four diseases, and they may ask due to the deficiency of what enzyme that diseases are occurring. Any kind of a question can be asked from the any kind of a corner. There is no point of getting confused over there. Everyone try to understand very correctly. Now, I will explain. You have to remember exactly the cause. Exactly the cause you have to remember. Lysosomal storage diseases. Everyone understand, friends? Lysosomal storage diseases are due to the absence of one or more lysosomal enzymes. What, friends? Lysosomal storage diseases are due to the absence of one or more lysosomal enzymes. Number one, everyone listen. Number one is called as acid maltase deficiency. Number two, we can call it Tay-Sachs disease. Very important for the exam, friends. Please don't skip, don't neglect my words. Number three, call it as the Gaucher's disease. Now, everyone, please listen. If you observe the acid maltase deficiency, you know, right? What happened to the excess glucose in the body? I believe everyone remember in the yesterday biochemistry class, in the last two classes of biochemistry we discussed the same. What happened generally, friends? In the body, excess glucose is getting converted into the glycogen when the carbohydrate resources are extremely full. I mean to say, when the glucose level are extremely higher in the body, excess glucose converted into the glycogen. And you know very well, during the time of a fasting or during the adverse condition, what happened, friends? Glycogen break down back to the glucose, correct now? But sometimes what happens, there is a mutation in some enzymes. Listen, everyone, there is a mutation in some enzymes. Due to the mutation in some enzymes, glycogen break down into glucose. Due to the mutation, glycogen is going to break down into the glucose. Sorry. If there is a mutation occur, glycogen cannot break down into glucose. Instead of breaking down into glucose, glycogen can store as it is. This kind of a condition only we can call it as acid maltase deficiency. We widely call it, friends, lysosomal glycogen storage disease. Very good. Which leads to the accumulation of the glycogen in the muscle tissue itself. Friends, there is no point of getting confused and it is very easy to understand. Next one we can call it Tay-Sachs disease. I'll explain. Why does this particular Tay-Sachs disease occur? Listen, friends, it is due to a deficiency. It is due to a deficiency in one of the enzymes. Listen, friends, in one of the enzymes which break down certain type of a lipid called as hexosaminidase A. Friends, please listen. This is the name of the lipid, hexosaminidase A. And what is the name of the enzyme? We call it as hexosaminidase. No need to get confused, friends. Name of the lipid is hexosaminidase A, and name of the enzyme that break down this particular lipid is hexosaminidase enzymes. Whenever there is a deficiency of this enzyme, what, friends? Whenever there is a deficiency of this enzyme, what generally happen? This lipid cannot break down. So instead of breaking down, what is happening, friends? These lipids are completely deposited or stored where? In the nervous tissue, which leads to the severe brain damage and cause the nervous degeneration. In most of the cases, friends, eventually it leads to the death at 3 years age only. Why this particular process is happening, friends? Tay-Sachs disease is a type of a disease which is caused by the due to the deficiency of an enzyme called as a group of enzymes called as hexosaminidase enzymes, which are involved in the breakdown of the lipid called as hexosaminidase A. No point of getting confused. The next one, Gaucher's disease. What is Gaucher's disease? It is due to the deficiency of a one enzyme called as the glucose cerebrosidase enzyme. I don't know whether you remember or not. We have studied when we are discussing about the plasma membrane especially. What is the major function of the glucose cerebrosidase, friends? It is required for the formation of glucose cerebrosides. What, friends? It is required for the formation of glucose cerebrosides. If there is a deficiency of this enzyme, what is going to happen, friends? This glucose cerebrosides never break down. Whenever they never break down, what happen? They will be stored in the long bones such as femur, they will be stored in the liver, they will be stored in the spleen. Whenever they stored in the liver and spleen, both the liver and spleen is getting enlarged completely and the bones starts becoming eroded completely. Correct now? And if this deficiency of this enzyme continues, friends, obviously it causes brain damage and it also causes the learning disability. You no need to remember the symptoms, friends. Remember what disease? It is caused by the deficiency of the what enzyme? That is the major concern. All right. Every one of you, please respond. Is it clear? The biggest organelle, lysosomes. Every one of you, please respond, friends. Okay, please kindly do not waste the time, friends. As I said, I kept the target 10th, but June 10th, but mostly by 8th and 9th of the June only we will complete the syllabus undoubtedly. Okay, so remember, the moment cell biology gets over and the moment biochemistry get over, we will start the test immediately. Hardly I will give you 2 to 3 days for the preparation, that's all. I'm reminding all of you right now only. I'll just give 2 to 3 days of time to prepare. So there will be a 50 to 75 questions from the cell biology, and there is a 50 to 75 questions from the biochemistry you people get in the exam. Okay. So make sure that you will cover the entire concepts very, very correctly. Okay. So friends, today is 10th. Okay. Today is 10th. On the 13th I will be releasing the MCQ book for the botany also, as well as the zoology also. I am sincerely requesting for those students who already paid the fees, kindly send your addresses to me personally in the WhatsApp. By that, we'll dispatch the MCQ book to every one of you on 13th morning. Okay, within 3 to 4 working days, you will receive the book. So by that, you can constantly focus on this practice. Okay, all right, all right, all right, all right. Okay, thank you all, thank you. So let's continue in the evening 4:30 p.m. Okay, if some people are unable to attend 4:30 p.m. class because some people are housewives and some are working women and working people, I encourage all of you at least go through the recorded without fail. It doesn't matter if you cannot attend the live or not, but make sure you watch the class. But give your best to go to the to attend the live. Okay. All right. All right. All right. Anveshana, you can, download the app. You will get the details. Okay. Thank you all. Thank you all. Thank you all.